Hello everyone, welcome to the BTC 509 bioinformatics tutorial again, and in this tutorial I'll be showing you BLAST for nucleotide sequence. It is almost similar with the BLAST for diamino acid sequence, but just to show you the BLAST for nucleotide sequence I am doing this tutorial. To do the BLAST you have to type the gene name first, say for our case XA21, and I'm setting it as nucleotide and then press search. Here I have to refine the results with Oryza sativa, as we know that this gene we are working with is from Oryza sativa, so it will take some time to filter out the results. So here is the sequence that we have, so we are clicking FASTA here. After clicking the FASTA, you can see that at the right side we have run BLAST option. We did this pre-primers thing in the previous tutorial, but now we'll be running BLAST for our nucleotide sequence. So I'm pressing run BLAST. So as always, you can paste your own FASTA sequence here, or for our case, it has already been pasted our entire FASTA sequence by this extension number. So we'll be doing BLAST, and you can also upload the FASTA sequence here by uploading your file that may contain your sequence, FASTA sequence of your nucleotide, and then just run BLAST. So this is the BLAST result, and the result prediction is almost same like this gene they have figured out is 100% identical to our given sequence, and this is 99.97% identical to our given sequence. So we will do one thing, that is we will check both the sequences, okay, and we'll see in which portion of the sequence is dissimilar with this particular gene. So we will be retrieving this FASTA sequence and this FASTA sequence, and we will check the genetic dissimilarities or the nucleotide sequence dissimilarities between these two. And to do so, we have to do sequence alignment. And one popular tool for sequence alignment we have already learned that is Clustal Omega. So after Googling Clustal Omega, you see the first result that came out. I'm clicking it here. I'll be posting the DNA sequence, so I'm clicking DNA here, and we will be pasting the FASTA sequence for these two. So this is the first FASTA sequence of our case, and then this is the second FASTA sequence for our case, and then just submit. So my job is currently running, and here is the result. And you see this sequence is not available in the second one. That means this is an extra sequence of nucleotide which the second one contains, and maybe there is some dissimilarity at the end. Yes, like I said to you that this sequence is not present in the first one. So that's it. That's how you can do multiple sequence alignment and see the similarities between two nucleotide sequences. One interesting thing I can do for you is that we can observe mutation from a sequence, okay, and to do so let's go to the input form again, and let's press the DNA here. What I will do is I will copy the sequence. Say I will copy this sequence up to this part. I'm pasting it here, and then I'm pressing enter. I'm pasting the sequence again. And say for an example, I have to change the number of this one, say like up to this one. I'm writing it at 32. And say I'm changing the sequence here at the, is written as 80. So I'm typing CC here, okay. I know where I have put the mutation, but say for an example, you were asked to work with two nucleotide sequence. Say for an example, coronavirus. The sequence for coronavirus is not same for all the countries, okay? And they do this kind of mutation in their nucleotide sequence. So if we want to figure out where the mutation occurs, you can just paste the FASTA sequence of these two variant of coronavirus, and then you just need to press submit, and it will show you the region where you have this mutation, okay. For our case, these two are the nucleotide base pairs that has this kind of mutation, as we put this mutation manually. So that's it for this tutorial. If you have any question, you can ask me in the next live session, okay. Thank you very much.